Welcome to this video. Today we are talking with Professor Marina Rodnina. Hello.
Hello.
You've just received the Albrecht-Kossel-Prize for your work on protein biosynthesis and the structure and function of the ribosome. Can you say a bit about your research and what fascinates you about the ribosome?
First of all, I would like to thank the community for giving me this prize. And I have to say that this is very important for me to get this prize. This was also an occasion where I started to look into the life of Albrecht-Kossel, and I realized that we have an almost forgotten pioneer of nucleic acids research. And this was a fantastic scientist who really solved many problems which at a time were absolutely pioneering, so made seminal contributions. But on the other hand, he was also a very modest, very nice person, doing a lot for the community, so it's a sort of a role model for all of us. So I highly recommend everybody to read about this man. I think this is a type of scientist which we need in the community more and more. Now, concerning my science, what I'm interested in is mechanisms, and actually I started to work on translation by chance. I was always interested in that, but I thought at a time that almost everything is solved, because at a time, and this was a long time ago, the main players were, or we thought they were, all found. But then when I started working on that, I realized that the mechanisms are all not known. And at a time, also the structures were not known. So this was an absolutely highly abstract field. So there were players, and then one could sort of build a story about that, but one couldn't imagine how they really work. So it was not a movie, it was a book, and it was a very short book. So what we started to do is to fill the players with life, and they started to move at the speed of real time. And then we started to solve the structures. And so in the meantime, we have, I have to say, colorful, although of course this is pseudo-color, so colored pictures of translation, how all the components interact with each other in real time, what they do, and how this at the end brings about protein synthesis.
And when you're talking about players, what do you mean with that?
Players are always ribosomes, and then the tRNAs, which bring amino acids to the ribosome, and then all these additional auxiliary factors which come and help the ribosome to make everything fast, accurate, and so that everything works properly for the cell. And of course also there are some regulatory processes, and this is what right now it's particularly interesting, is to see how this is regulated in the cell, because there's a basic fundamental program which of course works, but there are also regulatory mechanisms where depending on the cell everything works a little bit differently, and this is the reason why we have different proteins in different cells, and therefore we look like we look.
Mm-hmm. And how do you research that? How do you find out what is happening in different cells?
So we are very mechanistic. So what we do is we isolate each and every component. We purify it to homogeneity. So these are really chemically distinct entities. And then we reconstitute this in a working system. So this is like having a watch totally disassembled, everything purified, and then we see whether we can recombine it together, and this creates a watch. If yes, then we're lucky and we learn something. If not, we have to try again.
And is there a reason why the ribosome is so difficult to study?
I don't think it's particularly difficult to study, because every new object is difficult to study, and then at some time you start understanding or actually know how this works. And then of course you still have to make your experiment, but usually this is just the beginning where you really don't know what you have in hand. And the more experiments you make, the larger is the field for the intuition, where you actually have a feeling in which direction this moves. So of course it makes it easier to make the experiments. On the other hand, the ribosome is simply very large. And this makes a problem, of course, because there's lots of proteins, there's lots of RNA. This can be modified. It can be differentially modified in different cells. So, and this is a level of complexity which is already very large. And then it comes then to different players. And these different players may be different in the cells. So, for example, tRNAs, we all know then, for example, that in bacteria there is one particular type of tRNA which brings phenylalanine to the ribosome. But then you go further and then you realize that in eukaryotes there's this difference. So there are different isoacceptors and there are different isodecoders, and this is all expressed differently in different cells, and this makes a level of complexity which is really daunting. So this is where we have to understand now how this is all together works and how this is regulated. And there is one level of complexity over the other, and so sort of a code hidden in a genetic code. So of course we know the genetic code. No, of course we know the basics. But we still have to understand how these secondary and tertiary codes are hidden or start to work in each cell.
And what has inspired you most during your career?
Oh, I think this is just the beauty of these experiments. Part of what we are doing, these are real-time experiments, and the way it works is you take two solutions. One of them is fluorescence labeled, and then you rapidly mix them in the machine, which is called a stop flow. So, and what we have in time, this is something which is a time course, it's a fluorescence or thread time course, and you see in time this developing. And in principle, if one likes that, this is so fascinating. You see in a real time how molecules interact with each other. So the fluorescence goes up and then go down, and very often these so-called traces are rather complex, and then comes the intellectual challenge to understand what this means. And this is a real play and real challenge, and this is something which is probably what science is about.
Okay. And what do you enjoy most about your job as director of the Max Planck Institute of Biophysical Chemistry?
No, this is a difficult question, because I mean one possible answer is that I enjoy the breaks from this job because this is in these breaks I can talk to my lab and we can discuss the projects, we can discuss their results. I really enjoy this. So on the other hand, being a director means that one also has a lot of power and a possibility to develop new concepts. And this is, of course, this is a totally different level, and this is a great thing to do. It's a lots of work, but this is also a possibility to change the things, to do something really new.
Mm-hmm. And how do you keep up with what's happening in recent research?
I'm referring quite a lot. I'm referring original papers, and I usually take most of the papers that I'm really interested in for referring. I'm also referring lots of applications for European Union, also in Germany. I have to say mostly international. So this is already a very important thing. On the other hand, I used to go to really lots of conferences, and since we have Corona, I realized how important that was for me. Going to a conference, just listening to the talk, and talking to people after that, this is really priceless. So this is we have to move from this Corona limitations and from the Zoom, which is very convenient, of course, because nobody needs to stand up from the chair. But on the other hand, we are missing all this discussion. So the discussion after the talk, discussion in the coffee breaks, discussion in the evening with the wine. We are really missing all that. And this is an inherent part of science, which is so important that we should really not miss on that.
And how do you think does this affect students?
This is a really dramatic story. I think the students really suffer corona for different levels. So first of all, they really don't have an access to this high discussion level in science. So all these 2 meetings, of course, they are tightly scheduled. You have 5 minutes for questions. You cannot go somewhere and ask a question. Of course, there are these breakout rooms, and everybody is supposed to go there, but in reality, nobody is there. And so particularly not people who really did the job on that. They are somehow spread. Nobody knows how to do that, and nobody has time also. So for the students, they are missing a very important level of doing science. They're also not traveling to other countries. They don't see how this is working there. They cannot go for a course or for doing joint experiments somewhere else, so that they would also see other countries. Go, for example, to the US, see how the US system is working, or to UK or to France. This is, in fact, it's very dramatic. And also they don't see their peers, so the discussions also with the peers are not sufficient. And we at the Institute, we are really trying very hard now to restore at least this culture of interaction with the peers, and also of again introducing the seminars as they used to be, regular and very vivid, but this is quite some challenge.
What else do you do when you're not doing science, or what do you do in your spare time?
Well, we used to travel quite a lot, and this traveling, this was one of the parts also of my job, which I really liked, because I like to go to different places, also visit different labs, but also see how people live in different countries and different cities. And this actually is not working anymore. So we used to go to the opera quite a lot in different places. This also didn't work. So all these cultural enterprises where we used to go, just going for the exhibitions and whatever, concerts, yeah, it's a difficulty. So the only thing which remained, this is actually reading. So I enjoy reading, and of course I do that as much as I can.
Thank you very much for the interview.
Yeah, my pleasure.